Hi everybody, we're now moving on to a video on enzymes. First, you can look at the slide. Yeah, good lactose intolerant cartoon. Okay, let's move on to a brief discussion of enzymes. And it might be curious to you why we're looking at enzymes at all, because you thought we were focusing on energy in this module, and we are. But we're going to need enzymes in particular for this next part. To break a chemical bond, and to release its energy. If you think about how do we get the energy out of a sandwich? How do we get the energy out of a carbohydrate? We're going to have to break the bond. And we can do that in a couple of different ways. We can heat up a substance, and enough energy in the system, if you think about it, sugar and water, for example, if you heat it with enough heat, eventually bonds begin to break. It kind of falls apart. That's not really an option inside of our body. They heat up food at high temperatures to break the bonds and to release the energy. Or option two is to lower the energy needed to make the bonds break. You may have seen diagrams like this before. This is called the activation energy figure. And what this is showing you here is if we said there's no enzymes available, we're going to need, if you kind of watch the marker here, we're going to need to have this much heat or this much energy to cause the bonds to break, to then release the energy and produce products. That's a lot of energy. If you look at, let me switch colors here, I'll go yellow, or if we have enzymes, all we need to have is this much energy. That is, if we lower the energy required, not very good drawing, but if we lower the energy required, we can break the bonds without a lot of excess heat. So if that's a little confusing, don't want to bother you too much, focus on this concept here. Focus on what's called activation energy. Enzymes make it possible for reactions to happen at lower energy levels, so they happen more often than high energy levels. So that'll be a little clearer in class. But again, see the two options here. We can either heat up something to make the bonds break, or we can lower the energy required to make the bonds break. And that's what enzymes do. They help us make the energy required to be less. This is just showing you a schematic of the metabolic reactions inside of a cell. And you can see that every dot on this chart, it's a little hard to see, is supposed to be a different chemical. Between one dot and the next, an enzyme is required to make that reaction happen. So you see how important enzymes are. A particular enzyme makes each of those reactions happen. And that is basically the energy diagram, or the chemical diagram, if you will, inside of a cell. The sum of all of the reactions we refer to as metabolism. You've heard perhaps of terms like anabolism or catabolism. We won't go there with that detail. Metabolism means all of your chemical reactions. Don't think about metabolism like I have a high metabolic rate, or I have a high metabolism or low metabolism, even though that's kind of what it means too. Metabolism really means all the chemical reactions that's required to make you work. And enzymes are required to make those reactions happen. So what enzymes do is they help speed chemical reactions, because they can make a reaction happen and yet they themselves are not changed. So you see here, they make reactions happen a lot sooner, or at a lot lower temperature, or with less energy than would have normally happened on their own. So that concept again of lowering the activation energy is primarily what enzymes do. They make it easier for molecules to break, or easier for molecules to bond together, which then makes the reactions happen at a rate fast enough for us to work. And as you see here, nearly every metabolic reaction requires an enzyme, and you see it over here. Please remember, enzymes are proteins. Proteins have very specific particular shapes. The little balls you see on here are trying to represent different amino acids. And the right amino acid sequence caused that thing to fold up into that shape. And that shape is critical for how it fits with other molecules in the cell. That'll be clear to you in a second. Each enzyme, each of these proteins recognizes one specific target molecule, and that's called its substrate. So here you see the enzyme this is the enzyme lactase. Okay, here is its substrate lactose. Notice it has a site here called an active site that, because of the shape, and let me go back for a second, pretend that it happens to be right over here. Okay, there's a particular shape in that protein, in that enzyme, that's going to fit this particular substrate, in this case lactose. So the substrate is going to bind to the enzyme, that particular enzyme, at its active site, and they kind of fit together. The term we usually refer to this as is lock and key, that one enzyme fits one substrate. Okay? Because of this, the enzyme kind of stresses or breaks or helps break that bond and out pop the two products. In this case, glucose and galactose are the two monosaccharides that make up lactose. Notice the enzyme wasn't changed, and it can do this again and again and again. Here's some examples of some substrates and some enzymes. Don't worry about what they do, but I want you to see the A's. Lactose is broken down by lactase. Cellulose is by cellulase. Proteins by proteases. So A's refers to an enzyme. This is just showing you another look at what's called the enzyme substrate complex. The substrate is the substance is the reactant. Okay, and here's the enzyme, here's the substrate, here's what's being acted upon, and they're going to join together. So if you see that term enzyme substrate complex, it refers to the fact that the substrate can fit and join into the enzyme. The enzyme sort of stresses the molecule and makes it easier for it to break. But because it's a protein, its shape is hugely important. If the shape was wrong, if the shape was not correct, the substrate won't fit into it, right? If the key is bent, it won't fit into the lock. If the lock is smashed, the key won't fit into it. So here's an example of, once again, lactase, the enzyme that breaks down lactose or milk sugar. If there's a mutation in that gene, and some people have this, then their lactase they make is of the wrong shape. And now it's not going to be able to fit. Lactose will not fit into their version of lactase, and that will lead to lactose intolerance. If they consume milk sugar, they get a tummy ache because that lactose does not get broken down very quickly, and it causes some digestive uncomfort. I'm just going to show you one more example of this. This is one we've mentioned in class. In proteins, there is a number of amino acids. In particular, one of these is called phenylalanine. If somebody lacks the PAH enzyme, that's going to be a problem. For those people who do have it, who are normal, who do not have the condition PKU, then their PAH is going to break down phenylalanine into harmless byproducts, and they're good. If someone has PKU, they lack this enzyme. So what happens to phenylalanine is it accumulates in their system, and it builds up to dangerous levels. And the people that are most prone to seeing the effects of this are those very young children who are still developing of the brain development. And so excess phenylalanine in these children, because they lack that enzyme, can cause PKU, phenylketonuria, which can result in mental retardation. So here is just one more example of, if they don't have that right enzyme in the right shape, then they're not going to be able to carry out the reaction properly. And let me remind you, not that I should need to, let me remind you one more time, where does the shape of a protein come from? It comes from the sequence of the amino acids. Where does the sequence of amino acids come from? That comes from the DNA. Okay. Just briefly, things that can affect how well enzymes work. Environmental conditions like temperature or pH or the environment. If in the cell there's the wrong pH or the wrong ion concentration, or if someone has a fever, it can affect the shape of the enzyme and therefore how it works. Cofactors and coenzymes. The reason we take vitamins, for example, is they can serve as coenzymes and can help enzymes get into the right shape to do their jobs. Zinc and iron and vitamins are again examples of this. Another example, iron, it's needed to have hemoglobin work properly to carry oxygen. And there's enzyme inhibitors. And I'll just show you a slide of this here. One of the ways that we control our reactions in the body is by actually inhibiting enzymes sometimes too. And I don't want to get too detailed with this, but this shows you here the normal substrate fits into the active site and we get the reaction. If we have an inhibitor that fits into that site and kind of gets in the way, now this substrate cannot get in there. If it can't get in there, there's going to be no reaction. So it just shows you how the body can regulate rates of reactions. If it produces inhibitors, it can slow reactions down. If it doesn't produce inhibitors, it speeds them up. And finally, the big finish. What should you have gotten from this video? Enzymes are very, very critical for our metabolism. They are the helpers, if you will, to help one molecule break and form another one, or help one molecule form another one. They are very specific in shape because they're proteins, and they can only do their job, only specifically do their job with a substrate that they fit with. Sucrase cannot break down lactose. Lactase cannot break down sucrose. They are very, very specific. If the enzymes are misshapen because of errors in the DNA, which result in errors in the structure, they're not going to work. The process of breaking down our food, and this goes back to one of our previous videos, we talk about breaking down proteins to amino acids or breaking down polysaccharides to monosaccharides. All those require enzymes. We talk about chemical digestion. We're talking about how do we chemically break down big molecules to small ones. And the process of using monomers, the monomers from the macromolecules and making ATP requires enzymes. That's sort of a wordy way of saying when we want to make ATP, we want to make the energy that we need to make our cells work, we're going to have enzymes to power that series of reactions too. That's it on enzymes. On to cell respiration in the next video.